Good afternoon, everyone. It is a pleasure for being here and for the slides. And first of all, I would like to thank Nestlé for putting together this symposium and inviting me at being part of this.
Can we ask, please, switch the presentation? Sorry for that.
No, that's...
We all aging, you know, and it's how it works.
Me too.
Yeah.
Yes.
Yeah.
Okay.
Okay. So now you know what to do. Don't worry. We're not that dimensional.
No,
So
that's okay. Sooner or later. Okay. So this is my presentation. Age-associated cellular decline. I'm a geriatrician as a background. I've been working in the field of preventing disability in older persons, and I struggle every day in the clinical practice with age-related conditions that are responsible for disability and functional loss that are really burdening the individual as well as the public health system. And it is needed to anticipate the loss of function, and for this reason we need to go into the details of the mechanism regulating the homeostatic disruption that is occurring with aging. And a special focus, I think, has to be devoted to the cellular mechanisms because they will leave us understanding what is going wrong in a very early stage so that we may potentially identify targets for future treatment. When we deal with mechanism of aging, we deal with something that is very complex and in which everything is true and the opposite is also true. We have no clear understanding about why we age. There are in literature more than 300 different theories of aging, and none of them is able to explain such a complex mechanism that is perhaps explained by a collection of them in a complementary way. And for sure, the longer is our life, the more complex is to disentangle the intricate network of mechanisms and understanding what is happening. For this reason, working on cells might be nice in order to understand what is going wrong behind the clinical manifestation of aging. In order to understand and try to structure a presentation about why cells decline with aging, I think this paper that has been published a few years ago in Cell may really provide some support. In this paper that has been, it can be considered a cornerstone paper in the field, authors have described 9 different reasons why our cells are aging. And I will provide in these 20 minutes an overview of them, focusing more carefully on those that are more related to sarcopenia, to frailty, not because of importance per se, but because to date the evidence is stronger in some of these aspects than others. First of all, the genomic instability. We know, and it's well established in literature, that the DNA damage is responsible to a certain extent to the aging of the individual, or better, that with aging there is an accumulation of DNA damages. And these DNA damages are caused by endogenous and exogenous stressors that are continuously soliciting our organism. And the DNA damage is also been indicated as one of the major reasons for the onset of cancer and oncological conditions, mainly when the cell has a certain level of DNA damage, may follow 2 different stories, may stop duplicating and replicating itself, so simply it ages and it is put to death by the organism, or it continues being replicated and gives life to cancer. It is not so easy, however, because if we look at studies like this, we see that the accumulation of deficits in the DNA occurs in a relatively early stage of life, and it is simultaneous with the reduction of stem cell division rate. That is, these are very premature phenomena that are occurring during our life, and the decline of the aging functions, as well as the increase of cancer incidence, occur later in life. So perhaps reading this slide, we may say that DNA damage, that the genomic instability may predispose the organism to aging, but they may not by themselves alone explain the entire picture. This is another well-known mechanism of aging, the telomere shortening. Telomeres are these caps at the end of chromosomes that are involved in the protection of chromosomes throughout life and throughout the multiple divisions that the cell may have during life. There are a lot of studies that have shown how telomeres length is associated with aging and health. Just yesterday in experimental gerontology, there was a paper that was showing how telomere length is able to predict health status and aging in horses, so also across species. Epigenetic alterations are also playing an important role in aging. When we talk about aging, but more in general in medicine, we tend to look at the individual. We tend to look at the status at this moment, forgetting the story of the individual and the story of the progenitors, which are important information to consider in the clinical practice, but also in the research field. This is, for example, a study that is showing how the lifespan and the health status of the offspring is predicted and is associated with the health status and life at the procreation of its father. That is, what is given us or to ourselves by our progenitors is playing a role at explaining the way in which we age and our cell age. This is a more complex and perhaps not so studied mechanism that is related to the quality of the proteostasis. The proteostasis is mainly the homeostatic mechanism that is regulating proteins quality and quantity in our organism. A disruption of this mechanism may lead to aberrant forms of proteins that may lead to the destruction and dysfunction of cells. This is another of the 9 mechanisms that have been indicated as hallmarks of aging. In terms of nutrition, we are here at the ESPEN meeting. I think one of the most important mechanisms of cellular aging has to do with the nutrient sensing. That is, all the organisms and our cells are always looking for food, for nutrients, and are always looking to increase the energetic, to improve the energetic homeostasis. This is something that is true throughout living species. And in humans and in animal models, it has been shown that a very strong role in determining the aging process is played by dietary restriction, energy restriction. Here you can see 2 studies in which in one is presented the okay, how the dietary restriction is able to affect the aging rate throughout species. On the other side, you see the effects of caloric restriction in a monkey. There is plenty of literature that is showing how this model might be extremely promising also in humans. Nevertheless, the application of caloric restriction has to be carefully debated when applied to humans, because for ethical reasons and also for cost effectiveness. Let's think, for example, at what means applying caloric restriction to an 80 years old person compared to applying it to a 20 years old person. Also the effects and the benefits of an intervention like this will be different, because the effects will be exerted over a different lifespan. One of the most crucial mechanism of aging has to deal with mitochondria. Mitochondria are these organelles that are often forgotten and are often ignored, but are really playing major different roles in the health of our cells and our bodies. Mitochondria are at the center of different mechanisms that are related to the stem cell function, to inflammation, to cellular senescence, can be somehow depicted as the power plant of our cells. When they work, the energy in our cells, thus in our organism, works. If there is a mitochondrial dysfunction, then we have a blackout. And the role that mitochondria play in our organism is well evident, and there is an increasing body of evidence in this field, also in humans. Here there are two examples showing how the mitochondrial dysfunction has been associated with major age-related conditions. On the left side, you can see a paper showing mitochondrial dysfunction at the basis of sarcopenia, that is the skeletal muscle decline in terms of quantity and quality of the skeletal muscle. Persons with sarcopenia are persons who have a mitochondrial dysfunction that may affect directly, indirectly, their function, their general physical function. On this other side of the slide, you can see another condition that is very frequent in older persons, but is often neglected and may worth more attention. Here it is shown the results of a study suggesting that the idiopathic chronic fatigue, a symptom that is very commonly presented by older persons, is strongly associated with the quality and quantity of mitochondria. Fatigue is a symptom that is highly prevalent in the general population. This is a study that was showing, for example, that 1 out of 3 persons aged 65 years and older present and self-report some complaints related to exhaustion, to physical exhaustion, to fatigue. So it's a symptom that is out there, is a red flag. When we have a person with fatigue, we know that that person may be at risk of frailty, of disability, but is also a symptom for which we do nothing. We have no interventions to propose. Now, if fatigue is the clinical manifestation of the mitochondrial dysfunction, we may have a clinical target for intervening in the future with a direct biological pathway to tackle. And the relationship between fatigue, aging, and frailty as accumulation of health deficit is also described in this figure. That is, these are secondary analysis from a study that we conducted a few years ago, in which, as you can see, persons with fatigue, with this, again, very common symptom in the outpatient clinical population, is a symptom that is associated with both aging and biological aging. Persons with fatigue are persons that are experiencing an accelerated rate of aging. So again, this may represent a target for potential interventions that to date remains there, burdening the quality of life of the individual and left there as a source of major expenses for public health. Coming back to the mitochondria, to those organelles, there are different ways in which mitochondria are controlled. Mitophagy plays for sure a central role because it's there are systems that are there regulating mitochondria, and the failing of them is also a risk factor for sarcopenia and skeletal muscle dysfunction. And as you can see in this graph, in this paper, persons who are pre-frail or presenting some physical impairment, early stages of dysfunction, have also a very specific biological signature focused on the mitochondrial dysfunction, the blue area in that network of biomarkers. And there are also indirect measures of mitochondrial dysfunction at the basis of age-related conditions such as multimorbidity. Here there is the demonstration that glutathione is not only declining with age, but is also associated with a different rate of multimorbidity onset development because potentially related to the mitochondrial dysfunction. There are two other mechanisms that are also part of the cellular aging, the cellular senescence, that is the way in which our organism put hand to the declining cells, and there is also the exhaustion of stem cells that are also other two mechanisms that are worth to be mentioned as part of those that are influencing the cellular aging. Another one, the last one of these 9, is related to the communication, the possible communication existing between cells. There are studies like this in which transfusing the blood of old mice to young mice provided a sort of acceleration of aging, and vice versa, transfusing the blood of young mice to old mice was providing a sort of rejuvenation for the older animal. This has led researchers at thinking that the possibility that there are some molecules, some substances in our bloods that are playing an important role in the communication across tissues and organs. And there are studies like the one on the left that are suggesting the garbaging. We have the inflammaging theory, for which the low chronic inflammation is responsible for the disruption of tissues or organisms and organs and functions. But I mean, there are all the debris, cellular debris, all the garbage that over time accumulates in our organisms because of the multiple replication and cellular divisions that we are experiencing in every moment during our life, that may create a sort of exhaustion and generate the organism dysfunction. Now, why we talk at the beginning of this symposium on frailty, on sarcopenia, on nutrition, on very clinically relevant aspects about cells, because we are, I think, in a moment in which we are transitioning the traditional care that is offered to our older persons, and is offered in a traditional standalone disease approach. We treat disease per disease. And we are in a phase in which we should look more carefully at the biology of diseases, but more importantly, at the biology of aging. Because after all, age is the main common, shared by every one of us, a risk factor for any kind of disease of old age. As soon as we age, we have a risk factor for age-related diseases. And understanding the mechanism of aging, so why we age, why our cells are starting failing in their work and function, may provide us some novel hints and promising tools for intervening at the very basis of advanced age illnesses. And this is a very promising field of research, and we are not really starting from scratch. Today, we know that physical exercise, caloric restriction, healthy diet, in particular Mediterranean diet, are able to improve our quality of life, our functions, and prevent age-related conditions. But these three lifestyle modifications are positively acting on our organism because there are some underlying biological pathways that are related to their benefit, to the effects of the interventions, but for sure are also related to the aging process. So understanding this mechanism may provide us a new way of treating the age-related conditions because identifying the biological substratum on which the traditional diseases are built on. So, in conclusion, the phenotypic manifestations of aging are extremely heterogeneous, but their manifestation is simply mirroring the complexity of the aging phenomenon that is shared by every living species, and that may provide the critical foundations for the next to come management of age-related conditions. To date, we need to really start looking at the aging population in a different way, quitting the traditional standalone disease approach, the obsolete model defining a person as sick on the basis of arbitrary decided cut points on very organ-specific mechanism, and trying to have a broader perspective, including the aging process as part of the pathological conditions. For sure, we are just at the very beginning of this process, so we need to identify those conditions. I've mentioned here fatigue and sarcopenia, but frailty is also another condition that may represent a benchmark for developing this new clinical practice. For sure, now we have to really research a lot in this field. Thank you.